From the American Society for Microbiology, this is TWIV, This Week in Virology, episode 555, recorded on June 22nd, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today we're coming to you from sunny San Francisco, California, where it's 24 Celsius. It's a really nice day outside. We're at ASM Microbe 2019. And as you can probably hear, we're on the exhibit floor where people are milling around looking at exhibits. Joining me here today, my regular TWIV host, Brianne Barker.
Hi, everybody. Good to be here.
Your first TWIV in vivo.
It is my first TWIV in vivo. I'm really excited.
It's good to have you here. You came to the meeting to check out microbiology and virology,
I
right?
came to the meeting to check out microbiology, virology, microbiology teaching.
Right.
There are lots of great things here.
You enjoying it so far?
I'm enjoying it quite a bit.
All right, good to have you. And we have today a guest host. Now and then we do that. He is from Penn State University and the Curiosity Podcast. Please welcome Calvin Yeager.
Hey, thanks for having me. I wish that we had Dixon here so we could try to figure out exactly what kind of parasite I am to be here and taking. But I guess that we could still figure it out.
I thought you're an ectoparasite. What do you right? For sure.
It's a pleasure to be here.
Thanks for joining us. And finally, our guest is from Penn State University. He's also the ASV president-elect to be, Craig Cameron. Welcome.
Well, thank you for the invitation. It's a pleasure to be here.
I was hesitating to think of what else I should say besides that, but you're going to be ASV president next month.
President-elect officially next month, but also I'm going to be moving to UNC School of Medicine in the fall.
Doing everything at once, right?
Yeah, all at once, right? That's the way
thanks.
to do it.
Thank you for joining us. So we're going to talk a little bit about Craig's career and his work. And I should say that Calvin is a PhD student in Craig's lab.
Yet another parasite once again.
And so this is an unusual little dynamic here. We'll see how this goes. And but first, I want to remind the audience you should stick around. No leaving now, okay? We have these cool T-shirts to give away. I'm going to throw some out. They are World Tour, TWIV World Tour T-shirts. And on the front is that cool graphic, which was made by Sharon Isern, who's a virologist at Florida Gulf Coast University. And she has a little science art thing going on the side. It's called steampunkphage.com. And you can buy those there. But we're going to throw a few out into the real TWIV fans here. The real ones. And how do you know you're a real one? We'll find out. We'll find out. So stick around. Okay, Craig, let's start with you. Let's cover your history.
Okay.
Where are you from originally?
I grew up in a suburb of Cleveland, Shaker Heights, Ohio.
Yeah, I know Shaker Heights. Nice place.
Yes.
You went to high school there?
Yeah, from elementary school through high school. Yeah.
And where'd you go to college?
I went to Howard University, majored in chemistry, minored in math.
So going in, you knew you wanted to major in science, right?
Going in, I thought I wanted to become an MD. I thought I really wanted to practice medicine, and the selection of chemistry as a major was kind of strategic. You know, a lot of applicants to medical schools majored in biology, so chemistry would set you apart. What I found in the first couple of years, though, going into the hospitals trying to do service. This was in the 80s, where AIDS was on the rise. I just couldn't deal with aging, death, and dying on a regular basis, and I had been doing research, and so my career path changed.
About when in college was that, second year?
It was about my second year.
So then you decided you wanted to do research. You had been doing some lab work and you liked it.
And I was a part of the Minority Access to Research Careers program that was funded by NIGMS. Mm-hmm. And so I had to do research during the academic year as well as in the summer. And so all of that experience kind of showed me that discovery at the bench was very exciting, and I didn't have to deal, with some of the other issues that you have to deal with when you're in a clinic.
What type of research were you doing then?
So I started out studying an enzyme, dopamine beta hydroxylase, so doing basic enzymology. But every summer, I spent at Standard Oil And then Standard Oil was bought out and became British Petroleum. So I was actually working in a biotech department at Standard Oil, we were sequencing a lot of organisms, ribosomal RNAs, so we could make tests for contamination in production wells in Prudhoe Bay. Yeah.
So at what point in college did you decide to get a PhD?
Well, I actually applied to medical schools too. So it wasn't until the very end that I actually made the decision. I mean, I did the MCAT and the GRE. I was kind of on the fence. And a lot of it, related to my family. My parents knew exactly what it was, to go into, a career path in medicine. But the PhD, what are you going to do? You're going to more school, what is the outcome of that? So, yeah, ultimately, I stood up for myself and I said, this is really what I want to do. And so I went to graduate school.
And where was that?
Case Western.
Cleveland.
Yes, back in Cleveland. Yeah.
Okay. And what did you do there? Who'd you work with?
So I worked with Jonathan Lise in the biochemistry department. Jonathan's now at Northwestern, but he was a retrovirologist. And again, that was really one of the areas of interest that I had in college because AIDS was just everywhere. And, this was before AIDS had a name, the virus had a name. And about at the end, it became pretty clear that it was a retrovirus. And so I wanted to study retroviruses, and Case had a really nice group of avian tumor virologists. Sinjin Kung, Jonathan Lee, Susan Payne was there, equine infectious anemia virus. So I thought, it would be a good environment to learn retroviruses. Yeah.
So I was looking at your papers, and I noticed some of your early papers in the 90s, I guess that would be with Jonathan, on retrovirus protease with Ann Skalka.
Right. Yeah, no, and so it's one of those things, that time in my history was a very exciting period, and I think it imprinted the way I do science now. So it wasn't just Ann Skalka as a part of this collaboration. It was also Alex Lodauer and Irene Weber. So right from the beginning, I learned the importance of, being interdisciplinary, doing structural biology, not forgetting the organism, going back into the cell, as well as doing the biochemistry. Yeah.
So I wrote Ann Skalka to ask about you.
Oh, okay. Hopefully she said nice things.
Because I work with her frequently on our ASM textbook, Principles of Virology.
Yes.
And I said, did you know Craig? And here's what she wrote. She wrote, I knew Craig as a graduate student of Jonathan Lise. During the 80s and 90s, my lab had a very productive collaboration with Jonathan's group and Alex Woldauer. We all worked closely together. We had numerous group meetings. You remember any of those?
Yeah, I remember all of them, yeah.
Craig played a prominent role in our work together on the avian ASV, avian sarcoma virus, and later HIV proteases. I remember Craig as very affable, bright, and dedicated. He was a valuable member of the team.
Nice.
So it's nice, right?
And that's what's left after the redacted portions.
Yeah, I didn't see that part. Couldn't get rid of it. Right? Right. Okay, so that was your PhD. So you finished in the 90s, I guess?
Yeah,
roughly?
93. And so I stayed on actually with Jonathan as a postdoc for a year. And that allowed me to do more biology. But then I moved to Penn State, to the chemistry department, to work with Steve Bankovic.
Okay.
And so I was in 94.
As a postdoc?
As a postdoc, yes.
Yeah, so you did a lot of pretty hardcore biochemistry.
Yeah.
And so you wanted to back off and learn some biology after that, yeah?
Yeah, no, so, I didn't have a lot of hands-on with the virology in the collaboration. So
Right.
it gave me the opportunity to learn how to do that.
So what did you work on at Penn State?
So, just a little background. As a graduate student, I spent a lot of time trying to understand the determinants of specificity for these viral proteases. And in the end, the strategy was, if I could convert the avian protease into the activity and specificity of the HIV protease, you might identify residues that would change in development of drug resistance. And during this process, every introduction of a residue from HIV into the ASV enzyme increased the activity. And back in those days, when you're doing a lot of site-directed mutagenesis, usually every mutation that you introduce reduces the activity. It doesn't increase the activity. And so this happened time and time again. And so what I wanted to do was to be able to learn, how to study enzyme mechanism in a way that I could actually tell people when they would ask, well, what have you done, to change the enzyme to make it more active? Just traditional biochemistry, would give you rates and maybe some information on specificity, but we weren't really looking at mechanism. So Steve Benkovic is an internationally renowned bioorganic mechanism type person, nucleic acid enzymologist, and so I went to learn from him.
All right.
Yeah.
Now, I noticed in 1991, you have a paper, seems to be the first one on the polio virus polymerase.
Yes.
And this is with Stuart Legreese on the paper.
Sure.
So how did that come? So you're still a student at the time, right?
Yeah, so Stuart was at Case at the time.
Right.
This was before he went to NIH, and he was on my committee. And Stuart studied reverse transcriptase. And so we just became fascinated by the work that he was doing, and there was a connection with Steve because Steve had begun to study the RNase-H activity of reverse transcriptase and the strand transfer mechanisms of reverse transcriptase. Yeah.
So that was your first foray into polio polymerase, which you've stayed with ever since,
Yes.
essentially, right?
Exactly.
Okay, so we're at your postdoc at Penn State. What's next?
So I started looking for jobs in '96, and I was offered a position in the biochemistry department at Penn State, in addition to some other places, but I chose to stay in the biochemistry department there at Penn State. So I started my faculty position July of 1997.
And that's where you've been.
And that's where I've been until today. Right?
So
I'm still there today.
You're still there, and you're going to move in the fall or something?
Yeah, so I start September 1st.
At
Microbiology and immunology.
University of North Carolina.
Yeah.
Wonderful place. I did 2 TWIVs there
Okay.
a couple of years ago because they have 14 virologists,
Yeah.
7 RNA virologists, and 7 DNA. So we did 2 separate TWIVs.
Nice.
There were 10 people in the audience for each one.
Yeah, no, I mean, it's a great place for virology, cell biology, bacteriology.
Should be fun.
biocosin-
Are you
yeah.
looking forward?
I'm very much so.
So you could be a chairman, right?
Yes, I will.
And you get to hire some new faculty members.
Yeah, we're gonna, the first goal is to create a strategic plan, to kind of see where the department wants to go, and then start hiring.
Good luck.
Thank you.
It's a very exciting opportunity.
Yeah.
Right? It's good. All right, so now back to science. Let's talk about a couple of topics that I think are pretty cool. And so there are 2 papers that are, I think, seminal that we should talk about. The first one is called the Broad Spectrum Antiviral Ribonucleoside Ribavirin is an RNA virus mutagen. So tell us what led up to this story.
So,
And Craig, a little higher with the mic. Thank you.
okay. So I started my lab with the mission of trying to understand the viral polymerase, RNA-dependent RNA polymerase, using polio as a model, with the notion that if we could understand the mechanism well enough, we might be able to tune biochemical, biophysical properties of the enzyme to create an enzyme mechanism-based strategy for viral attenuation. And so to do that, you really needed to be able to understand the single nucleotide addition cycle. And most of the studies of the viral polymerase, RDRPs, that had been done up until that point looked at the polymerization of radioactive nucleotides. And so you weren't looking at one nucleotide addition at a time. And so we developed those early assays for looking at single nucleotide addition. And we were approached by Schering-Plough. At the time, Schering-Plough had cornered the market on the therapeutic for hepatitis C. It was a combined therapy with ribavirin and interferon alpha. And this was about to come off patent. And so they wanted to replace the ribavirin portion of that cocktail. And the thought was, at the time, that the metabolite that was active, so ribavirin is a nucleoside, gets into the cell, it's converted to the monophosphate, but then to the di and triphosphate. But everyone thought that the monophosphate was the active species, and that it functioned by changing cellular nucleotide pool concentrations by targeting a metabolic enzyme. But the metabolite that accumulated to the highest level was the triphosphate. And this is, of course, looks just like a substrate for a viral polymerase. And so we had the opportunity to ask the question of to what extent would a viral polymerase utilize the triphosphate? Was it an efficient substrate? What was the specificity? And so ultimately, what we were able to show was that it was incorporated, utilized by the polymerase, but ambiguously. So it was templated by two nucleotides, cytidine and uridine, instead of just one. So it essentially functionally introduced errors into the genome.
So if you add ribavirin to a polio-infected cell, you will get a number of mutant viruses. Does it decrease the infectivity as a consequence?
Yes, as a consequence, yes.
So that's from the part that says it's an RNA virus mutagen,
Yes.
right? And that's the first time that had been figured out. Right?
Yes.
Because I remember back when I was a student, people had all kinds of ideas of how it worked, and nobody really knew.
Yeah, and people targeted were thinking cellular enzyme as a target because it had such broad spectrum activity.
Yeah.
But people always talked about the monophosphate. They really never talked about the triphosphate. So, once you begin to talk about the triphosphate, clearly all viruses have polymerases that could potentially utilize the compound.
So this was done with poliovirus, but is it also a mutagen for Hep C?
So that's where the controversy began. Right? So there were about 15 years of it is, it isn't. It is, it isn't. I think that right now most people believe that it does have mutagenic activity, but for some reason or another, people continue to look for alternative mechanisms to explain the antiviral activity. Now we're in the era of Harvoni, so those arguments have disappeared essentially.
Yeah,
So,
exactly.
And is that just the case with Hep C, or is it broader in other RNA viruses
I
as well?
think that, so it was one of those things. I think a lot of the debate with hepatitis C had a lot to do with positioning of different companies, right? Where it was an act of purely academic interest, most people came down on the side of the mutagenic activity. Yeah.
And so how good do these nucleosides have to be? Can they take things down to nothing, or is it just the carrying capacity of the virus that they need to start interfering with just a couple of logs worth of titer?
Yeah, I mean, a log or two is usually sufficient to begin to see the immune system in vivo start having a huge impact on the ability of the infection to be sustained.
All right, and the second paper, which is another big one, it's one of the ones I also teach undergraduates, RNA virus error catastrophe, direct molecular test using ribavirin.
Yeah. So like the first study, which was done in collaboration not only with Sheeran Cloud, but with Raul Andino's lab. Shane Crotty was a graduate student in his lab at the time. Shane was very interested in Eigen's, error catastrophe theory. And by virtue of being able to recognize ribavirin as a mutagen, it gave Shane the ability to actually ask to what extent mutagenesis of the genome would correlate with fitness reductions. And he could essentially show that if you incorporated enough, had enough mistakes introduced, it was lethal to the virus. Yeah.
And so explain the error catastrophe, the threshold idea to us.
Right. So the original error catastrophe hypothesis really was put forward by Eigen, and this was a situation where we all know about the consensus sequences for viruses, right? And when you look at a population of viruses, you're going to see single nucleotide changes relative to the consensus in the population, but you will always find the consensus in that population as well. Error catastrophe is a situation where the mutational load is so high that you can't find the consensus in the population any longer. And at that point, the fitness is thought to be reduced. So there has been, a debate about what we should call this antiviral therapy that's based on mutagenesis. we're not necessarily forcing viruses into air catastrophe, but we're trying to increase the mutational load to the extent that it's lethal for the virus. So we call it lethal mutagenesis, so that we stay away from a lot of the those like Eddie Holmes who are really words matter.
So if I remember in that paper, you showed that by treating infected cells with ribavirin, you only increase the number of mutations a few fold, but
Sure.
infectivity is gone. So
Right.
RNA viruses are pretty near the level of that error
That threshold, yes.
Whereas in other papers, people have shown that for DNA viruses, they're way below it, right? They exist orders of magnitude below the threshold.
Right.
Because they have error correction,
Exactly.
right?
And
mechanism.
that's really the fundamental difference. Yeah.
Right. Now, part of this is Fidelity, which I want to get into. But before I do, you mentioned antivirals based on this concept of pushing a virus over the threshold. Can I use threshold?
Yes.
Error threshold?
Yes.
Has anyone got such antivirals? Have they? I know that people talked about them for lots of different viruses, including HIV. Has it ever worked?
So there are a lot of companies that the basis of which has been creating what they've referred to as stealth nucleosides, and all of these have mutagenic activity. None of them have been approved, but they do exhibit antiviral activity, in the laboratory and in preclinical models. Yeah.
Do they do that specifically to a polymerase? Could you target a reverse transcriptase instead of an RDRP effectively by using nucleosides?
Sure, and one of the companies that formed, that was the target, HIV. It was an HIV therapy, yeah.
So these are all RNA viruses because they're so close to the threshold that this strategy would work.
Sure.
Right.
But it's one of those things. I mean, it could be possible to use this for DNA viruses. Every nucleotide that's incorporated is not necessarily excised by the 3' 5' exonuclease with the same efficiency as a normal mispair. So you can actually mutagenize DNA viruses too, but you have to think about the excision process as well.
Got it.
Yeah.
So a lot of this is related to aspects of polymerase fidelity.
Yes.
And my students always ask me a lot of questions about fidelity and why polymerases make so many mistakes. I usually talk about it being evolution related, and they come up with all these ideas of what if we had a perfect polymerase? So could we have a perfect polymerase?
Yeah. So you have to think about it from many perspectives. So if we think about the RNA viruses, now I think one of the hottest areas relates to all of the modifications, right? Methylation of the RNA that's happening. If you have a perfect polymerase, it might not be able to deal with the challenges of post-transcriptional modifications of the RNA. You have oxidative damage that happens. And so if introduction of 8-oxoguanine into RNA stopped the polymerase, and it probably would with the most faithful enzyme, you're now going to have a severe fitness consequence in a natural setting. So I think a lot of what we refer to as the low fidelity is there to kind of deal with the evolving nature of the genome and the substrates even.
That's a great point, the modifications. I hadn't thought about that.
Yeah.
In fact, last year here we had Stacy Horner on Twitter.
Oh, sure. Yeah,
She talked about methyl modification of A's.
Right.
And many people are interested in that. And so that might stop a polymerase.
Yeah, we haven't published this, but we've shown that, it does matter.
So does making a polymerase have lower fidelity deal with that then? Can you show that actually?
Well, you can bypass more efficiently, but then there are other consequences.
Okay.
So there is a balance, there is an optimum.
So to follow up with Brianne, could you make, if you had a perfect template with no modifications, just synthetic template that you looked at, could you make a perfect polymerase that never made a mistake?
We've never done it.
You've tried?
Yeah, it's one of those things. You select for high fidelity, and there are ways to do that, as we've done with Ribavirin, and we've never selected a perfect polymerase. I mean, in the case where we thought we had gotten something that was close to perfect, actually what we found was that we had created a derivative that was very, 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 very efficient at selecting against this analog that we were using in our screen, but it didn't change the overall ability to kind of take in, do the promiscuous incorporation, GU mispairing and things like that. So
RNA viruses don't have error correction, and so they exist near this threshold of mutagenesis, of lethality, say. DNA viruses have correction. That's why they exist way below it. Why the difference in the two kinds of replication strategies? Why would you have one exist near and one far away? Do we understand that?
I'm not going to say we understand it fully. Mm-hmm. I think when you think about how genomes have evolved, and polymerases have evolved, just for DNA to be, the heritable unit, we don't want to make mistakes. So those polymerases existed to have exonucleases and mismatch repair systems. With the RNA viruses existing as a population, actually provides, I think, increased fitness, right?
Yeah,
because
sure.
you can evolve very quickly in changing environments because you have a population that evolution can act on. So, so being too faithful in your sequence could create a bottleneck, if you will.
Yeah. So that makes sense. The RNA viruses have evolved to take advantage of this error prone by using quasi-species, as we used to call them. Now they're mutant swarms or something else. I still like quasi-species, although it's very wrong, right?
Mm.
It sounds good. And they, as you said, what's that?
It's fun to say.
Yeah.
Great to say. Students, undergraduates like it.
Undergraduates really like it.
Because I say the old term is quasi-species and the new one is mutant swarms, and they say, no, we like...
No, they give you quasi-species on the exam.
And that's important for them being able to adapt to new hosts, new environments, et cetera, right? And
Yes.
as we all know, RNA viruses rule the planet, right?
Exactly.
And that's one of the reasons. That's one of the reasons that they do. So let's talk a little bit about this fidelity property, and this starts with a really interesting 3D Pol mutant made by Raul Andino and Carla Kierkegaard, which was really selected for resistance to ribavirin. Tell us about that.
Yeah, no, so it was amazing. So all of our labs actually identified this one substitution in the polymerase that didn't make it resistant to ribavirin, but the virus became less sensitive to the drug. And ultimately, doing some biochemistry, what we were able to learn was that it really was a more faithful polymerase. And the way that it worked, it was in a region of the structure where you had the amino terminus kind of pinning down the active site.
Actually, we have a picture. If we can put up that image. I brought an image for you, Craig. I thought you would like that.
Nice. Yeah. So this glycine, when it's a glycine, the amino terminus will actually interact with the backbone of this glycine. I guess this view doesn't really necessarily show us exactly that conformational capacity, but the amino terminus buries itself into the active site, pinning down the active site. If you convert this glycine to a serine, you don't get that interaction. And so what ultimately happens is that the most efficiently incorporated nucleotide is always going to be in a correct base pair with this high fidelity variant. Everything else is just, really, it can't create the architecture of the active site that's necessary. So it's the fidelity decrease is not deliberate, if you will. You're making so many dynamics that if you have an incorrect base pair, catalysis won't go. just because you can't get the right architecture. So it was a very lucky mutant, and really for polio, no one else has really achieved a ribavirin-resistant phenotype with other than using the G64S.
So even though G64S isn't directly within the active site, it's still affecting the active site for that fidelity?
Yeah, so the dynamics of the active sites controlled by not only active site residues, but remote sites. And so G64S is connected dynamically to the amino terminus, which is then connected to the active site.
So on this picture, the active site is shown in yellow with the two aspartates, right, as balls.
Right.
And so what you're saying, and that's where the nucleotide is put into the growing chain, right?
Exactly.
And you're saying that the glycine can actually touch that, even though it looks far away here, it can actually insert near it, right?
No, so the amino terminus will get close to the active site,
Right.
And it has contacts with the glycine. But if you actually look at the molecular motions of the enzyme that are required for catalysis, this G64S substitution completely causes chaos in all of the dynamics globally.
Mm-hmm.
So it's almost like flexibility issues?
So it's allostery,
Okay.
right? So you regulate the active site and connect the active site to remote sites by conformational dynamics of the enzyme.
So that single amino acid chain makes the virus less sensitive to ribavirin, right? And as you said, it makes a more faithful polymerase. It doesn't make it completely perfect, right? But makes fewer errors.
It makes fewer errors, but it incorporates correct nucleotides at a slower rate.
So the incorporation rate is slower.
Yeah, about threefold.
Okay, but in cell culture, that doesn't matter for this virus. It grows normally, right?
Well, it depends on how you do the experiment. If you're doing a one-step growth curve, you don't see a difference.
Right.
But as you begin to, do additional experiments, that we're doing today, you can begin to see a fitness consequence.
Okay. In cell culture.
In cell culture.
Because I know in animals there's a big
Yes, it's
consequence.
attenuated in animals.
And if you do competition experiments, it loses all the time,
Yes,
right?
yes.
Right. That's an amazing mutant. This is a question I ask my undergraduates. Maybe you could take this one too.
I'll take it.
Okay, so you have ribavirin, which is a mutagen, and you make a resistant mutant, and they don't know the literature, right? So this is all new to them, right? You make a resistant mutant, and the drug still binds the polymerase, because I don't want them to think that the resistance is just so the drug can't interact. So what's the mechanism? And of course, the answer is it's improving the fidelity, but nobody gets it.
Yeah.
Right? It's a good question, isn't it?
Yeah, no.
Because that's the answer. But it makes you really understand what's going on here. Do other polymerases have such, and other viruses have such mutations that increase fidelity?
So I would say most of the RNA viruses that people have evaluated with ribavirin, actually, they're all sensitive. Mm-hmm. So within picornaviruses, it ranges the sensitivity, and so does the intrinsic fidelity of these polymerases. So,
Okay.
We've used Coxsackie B3 as another model to kind of compare to polio, and it's definitely more faithful
Right.
than polio, the polymerase.
Does that have anything to do with the need for a primer?
Well, they both need a primer, Coxsackie and polio. The fidelity differences I don't think are related to the use of a primer.
Okay, but with other viruses that don't need a primer?
With other viruses, if you're not a primer-dependent enzyme, you're going to be initiating de novo. Mm-hmm. Okay, and so it's going to be more difficult to utilize ribavirin and analogs in general at that initiation step. You really only see it happening at the elongation step. There's a stringency on the selection of nucleotides that you use for initiation de novo.
Okay.
So would you say that the polio polymerase is a low fidelity enzyme?
So yeah, when I first started, I mean, this was really one of the questions as a nucleic acid enzymologist that I really wanted to address. When you look at the fidelity of the RDRP, it's on par with the fidelity of any nucleic acid polymerase, replicative nucleic acid polymerase that lacks a proofreading mechanism.
Okay.
So yeah, it has not evolved to have a low fidelity.
Okay.
Yeah.
All right. Now, as you just mentioned, the polymerase is primer dependent.
Yes.
And in fact, for the picornas, VPG with two nucleotides is the primer, right? For other RNA pols, though, you don't need a primer. And so, like Hep C, right?
Well, yeah, you create your primer in situ, right? De novo,
Yes.
right? Yeah.
The polymerase makes a little shoulder, which that first base can abut to, right?
Right. And so, yes.
Is that more or less right? You could tell me if I'm wrong.
Yeah, no, I mean, so you begin to, you recognize the end of the template.
Right.
And you begin to lay down nucleotides. From a dinucleotide, you go from a dinucleotide to trinucleotide, tetranucleotide. You make your own primer, as opposed to coming in with this peptide primer and extending that tyrosine, tyrosine. Yeah.
So what I would like to know is why do we have these two different mechanisms? I would think that, so from my view, having this shoulder in the polymerase, that has to move out of the way at some point so that the chain can then move through. So that seems to be a lot of extra work. But is having a primer also equally disadvantageous, and that's why they both exist?
No, I mean, I think it's, just the way biology works, right? I mean, so there are many solutions, to the same problems. And, when you look at the lineage of polymerases, there are those that are de novo, that function de novo, are from a different lineage evolutionarily than those that can function like the coronaviruses and use a primer. So, so there are a lot of primases that are out there that function more like these de novo initiating enzymes and DNA polymerases, reverse transcriptases, always use a primer. In the case of retroviruses, the reverse transcriptases is the tRNA.
Right.
Yeah.
We were just talking on TWIV about these new DNA polymerases that have been found that are primer independent. The replicative polymerases, right? They apparently copy whole genomes, but they don't need primers. And there seems to be a trade-off because there are a lot of stops and starts, kind of like Pol2, right? It takes a while to get going. Those first few bases you put on, it's kind of a wobbly complex.
Yeah, just the stability of the duplex. Yeah.
And then the polymerase falls off a lot, but once it gets going, then it's racing down the highway.
Right.
And these primer-independent polymerases, which have been found in a phage.
One was a phage, and the other was in...
mobile genetic elements.
Yeah, it was in a transposon.
They also seem to have problems early on getting going. They fall off a lot, like the Pol2s.
I don't know about these, actually.
Yeah, they were just published recently. Really interesting. And, it brings up the question whether... And maybe that's why DNA polymerases evolved to have separate primers and replicative enzymes, right? Because
so, primases initiate de novo. I mean, the issue with the DNA polymerases is that the replication of the end problem. And that's really one of the reasons that, you don't want to... You need to be able to create the primer de novo.
Right.
And so you need that primase. And then, we have the telomeres now, right? Or repetitive sequences at the end. But for RNA viruses, it's one of those things. I think that one of the advantages of having protein, a peptide, at the 5' end relates to the fact that when you initiate de novo, you have a 5' triphosphate that's at the 5' end, and we've evolved innate mechanisms, right, to recognize that 5' triphosphate as non-self. So you turn on innate immune pathways. So an advantage for coronaviruses and having that peptide is that they don't have to worry about if that 5' end is displayed like you do with a flavivirus, for example, where if you don't get that 5' end capped before it's exposed and sensed by IFITs and rig I, and then you're going to have an issue. So the coupling, right, the processing of the RNA and timing thereof becomes really important.
That's a really cool insight, right? So if you don't use a protein primer, you need to cap the RNA right away, otherwise it's going to be sensed.
Right.
So if you don't cap newly synthesized RNA, do you have a triphosphate there?
Yes.
Is that okay.
Exactly. And that's one of the reasons that with the flaviviruses, when they mutate the capping enzyme, it's attenuating for the virus.
Right, because they're being sensed and eliminated.
And that's the importance of things like cap snatching with the orthomyxoviruses too.
They take a cap, they can't do it themselves, so they steal it from the host cell. This is great. It's just great, right? I love seeing the big picture like that. So this whole field of nucleic acid synthesis, as you know, used to be robust for DNA, and hardly anyone does it anymore. You're one of the few people that's still doing it for RNA. I think it's remarkable that you have your entire career you have pursued and continued to do this, because you're probably one of the few, right?
Yeah. No. Well, I mean, it's one of those things. I mean, we've been able to create technology in collaboration with others to understand better mechanism, and I think that our ability to understand the fundamental chemistry that's occurring at the active site of these enzymes will enable us to take advantage of that knowledge and create vaccines. I mean, so this really has been. We don't know where the next outbreak is going to come from, but if it's an RNA virus, it's going to have an RNA-dependent RNA polymerase.
Right.
And so mechanism-based platforms for attenuation, I think, could be very valuable. So that's really been at the heart of our motivation to learn as much as we can about these enzymes.
I think that's really important to point out, because you start saying we want to understand the polymerase, what controls its fidelity. You make this mutation, G64S. Everyone says, well, what's the interest? But now I understand people are introducing that into other picornas and trying to make vaccine strains.
Right, exactly.
So it's brilliant. You go from the basic science to therapeutic options.
Yes.
And that was probably hard to see in the beginning, right?
Yeah.
You can't predict that, where it's going to go. Now, in the years that you've been working on RNA synthesis, a lot of structures of RNA polymerases have come out. So I guess these have been incredibly useful for you, right, to see these?
Well, I mean, they've shown just how relevant the poliovirus model is, right? I mean, the active sites superimposed within tenths of angstroms, RMS deviation, right? So what we learn about the regulation and mechanism of nucleotide addition in the poliovirus system, it applies, and we've shown that practically too now. By looking not only at other picornas like Coxsackie, but looking at flavies like Zika and dengue, West Nile. Yeah.
So these polymerases, all the polymerases are right hands, right? They look like a right hand. You have an active site and fingers and palms. But from my understanding, the picorna polymerases, the thumb and fingers touch.
Yes.
So it's a closed active site. So why is that as opposed to the others?
So we learned a lot about the reason for this from just the studies that have been done on reverse transcriptase. Okay, so reverse transcriptase doesn't have the touching thumb and fingertips. Engaging the template and maintaining processivity on the template requires an interaction of the thumb in the major groove of the nucleic acid and the fingers interacting with the duplex. So if you have the flexibility of the fingers relative to the thumb, you're going to be more of a distributive enzyme. You're going to dissociate more often because that dissociation is driven by the movement of the thumb relative to the fingers. When you have that completely encircled active site, that fingertips-thumb interaction, that's more stable. So once the enzyme has engaged nucleic acid, it can go a greater distance without falling off.
Just more processive.
It's much more processive, but it's very, it's you can't understate the importance of this. So you take a lot of time and you introduce a lot of factors in your genome to orchestrate the initiation event, specifically on the genomic RNA. So you want to be able to, after you've invested all of that, energy and initiating, you don't want the polymerase to fall off before it reaches the end, if you can help it. Now, if it does fall off, you want it to be able to fall off in a way where transfer to a second template is driven by the nascent RNA, perhaps, in a homologous recombination event, not by a new, initiation event. So I think that's really one of the reasons that these RDRPs, they are extraordinarily processive. Thousands, we've been able to show, even with a double-stranded template, they can go thousands of base pairs using the strand displacement activity and not fall off.
So a double-strand template, the polymerase is just separating the two strands?
Yes. Yeah.
So the template's coming in through a hole on the polymerase, and does one strand not go in? The double strand doesn't go in, right?
Yeah, presumably the nascent RNA is coming out of channel. Yeah, and that's, we really don't have structures of these, of a real elongating enzyme.
People are working on it, I guess. Right?
Well, now with cryo, I think it's going to be more, we will realize that.
Yeah.
Trying to get a crystal
right.
of all of, that has been very difficult. But,
Right.
but yeah, we and others are now, with cryo, I think it's going to be realized.
So you mentioned recombination. I wanted to talk a little bit about that because recently you've gotten interested in what regulates recombination. So tell it, what is in these RNA viruses with one genome, what is recombination?
So, believe it or not, we've been interested in recombination from the very beginning. So, as a postdoc, that's what I studied when I studied reverse transcriptase. I was studying template switching. It's an integral part of the mechanism. You start with a tRNA at a recessed position on the 5' end of the template, and then you go to the 5' end, and then you have to transfer that from one end to the other. And for all intent and purposes, when we started, RTs kind of looked like RDRPs. They have the same active sites. And so in developing our primary extension assays, we were able to develop a recombination assay, but our attention was diverted because of all this Fidelity work and the Ribavirin and everything. But then later, we became more interested in recombination because of this observation that the site for extension, for adding the two uridyl residues to the peptide primer, that happens at an internal position in the genome. So you now have to do what is similar to a forced copy choice recombination event from that internal position to the 3' end. And it has to be directed by this 2 nucleotides extended peptide primer. And so that really was one of the reasons that we started thinking of, we developed a lot of the biochemical tools, but then Raul's lab wanted to identify a recombination defective mutant, and they had a screen for that. David Evans' lab in the UK created a very robust in vivo assay, and so it allowed us, the timing was just right for us to start thinking about this again. We were able to combine, these new derivatives with the biological assays and the biochemical assays that we had never reported to really begin to understand what the mechanism of recombination was. Yeah.
So you're capable of jumping onto that acceptor RNA with the polymerase just from, do you say 2 nucleotides worth of exposed RNA? What is the requirement for being able to hop onto that acceptor RNA?
So when you do a template switching event from the internal position of a genome, that's called copy choice. When you do a transfer from the end of a template, that's called forced copy choice. Okay, and so forced copy choice is very useful for helping to repair genomes. So RNA is very sensitive to damage. So we think that's one of the reasons that recombination is so important. But when you get to the end of the genome, 2 nucleotides is sufficient to actually recruit an acceptor template, and for that primer to now use that template and continue synthesis. And so, again, we're hoping that cryo will give us some structural information on this, but biochemically, 2 is sufficient.
That's interesting. You say that recombination may be in part driven by overcoming damage to the genome. It's kind of like why we think retroviral genomes are diploid.
Sure.
Then you do copy choice and you eliminate the damaged parts.
Right.
And so that's an interesting way of looking at it. So you can identify sites in the polymerase that regulate recombination, right?
Yes.
And they're distant from the active site.
Yes.
So how do they regulate recombination?
Again, a lot of it is dynamics.
Yeah.
So the size of the nucleic acid channel is
Okay.
regulated by a lot of these residues. So the ability to actually recruit the acceptor template, it requires a certain dimension of your nucleic acid binding site. And if you have a really tight binding site, you're not very proficient at
I see.
combination.
So when you make viruses that are less able to do recombination, how do they fare? Are they less competitive?
Yes.
And
Yeah. So all of Raoul's mutants are attenuated. The few that we have are attenuated. But there's complexity to this because every parameter, biochemical property that we've measured of a polymerase contributes, so fidelity, speed, all of these contribute to work combination efficiency, at least in vitro. So when you start trying to point to recombination alone as the cause of the phenotype that you're witnessing in an animal model, it might not be so, right? I mean,
Yeah. But I guess you could do an experiment where you would, say, mutagenize the genome, and under normal levels of recombination, you would be able to rescue. But then if you use the recombination deficient mutant, you would have less infectivity. Right?
yeah,
You
and,
can-
and that's kind of what people have done.
Yeah.
So Raoul and David Barton, actually, UC Denver, he's been able to show that the challenge of polio with ribavirin, the sensitivity to ribavirin goes up if you decrease the ability of the virus to recombine.
Makes perfect sense,
Yeah.
right? No, that's neat. All right, let's switch gears for our last topic here. Recently, you've gotten interested, and maybe I'm wrong on recent again, but you'll tell me. Recently, you got interested in single cell virology,
Yeah. Yes.
which, you know, I remember a paper or two from Andrei Lvov in the 50s where he picked single cells with a glass pipette and put them in, tried to infect them. But now you do it high tech. Tell us about that.
Yeah. So a lot of this work. I mean, it started maybe about 9 years ago, but inspired by the observation that we could make fidelity mutants in vitro, right? In cell culture, you see no phenotype. You go into an animal model, and these viruses are attenuated, right? Ultimately, the insight came with one of my graduate students, Sherry Lee. The quality of the genome only matters under conditions where you have single genomes or a few genomes being introduced into single cells. And so we created kind of like an artificial experiment where we looked at limiting dilution with a virus population and asked if you were wild type, were you robust to establishing infection or more robust than a fidelity mutant? And that's essentially what we saw. So these bottlenecking events that we would see in vivo were best mimicked under conditions where you had single cells being infected by single viruses. And so we embarked on a program to try to develop strategies to be able to do single cell analysis of viral infection dynamics in a robust way. And so we use microfluidic devices that have thousands of chambers, and in each chamber there's a trap where we can infect off chip with a virus that's expressing a fluorescent reporter, introduce the population of cells into the device, and you have one infected cell per well. And now you can monitor on the stage of a microscope that's jacketed to maintain humidity and CO2 temperature control. 30-minute intervals over a 36-hour time period, the dynamics of the infection in each well.
So,
and we're learning a lot from this.
so if you just do growth, let's say you measure the production of infectious viruses in thousands of single cells, what do you see?
So when you think about, so let's compare this first to what we would do what we would see if we did a population level typical cell culture experiment, right? You're gonna see an eclipse period, you'll see exponential growth, and you'll see the end, right? So what is that telling you? There's heterogeneity in the viruses. There's stochastic gene expression in the cells, right? So you're not going to have every pattern recognition receptor expressed in every cell. There are going to be proviral factors in some cells. So the first thing that we see is all of this heterogeneity. Every outcome, whether it be the dynamics, endpoints, are different. What we also learn is that if you just take the unadulterated average of the infection dynamics that you see, it superimposes with what you see in the tissue culture dish. But now, if you begin to evaluate parameters like the time it takes for a cell to establish an infection, being able to have detectable fluorescence, you see a distribution of times where half of the infections are beginning at 8 hours post-infection, when your one-step growth curve tells you that all the infections are completed by 8 hours. So you begin to see events that are masked at the population level. And so we're teasing out a lot of different aspects of even the interaction of a virus population with drugs. Not each cell can metabolize a drug in the same way, right? Or accumulate the drug to the same level. So you see very heterogeneous responses when you're doing the single cell analysis as opposed to doing the population level experiment.
Are you able to do sort of competition types of assays in that as well?
So that's one of the things we're going to talk about tomorrow. We're just beginning to do this. So when we've always used Coxsackie as a model. And when you look at their one-step growth behavior in HeLa cells, they superimpose. But when you now do the single cell analysis, you begin to see that Coxsackie is much more fit. It's a much better virus. It replicates to higher levels and faster in these same cells. And so that inspired us to begin to look at competition. And so you can see cooperation within a species, but you see strong competition between species using this device with really nice resolution. Yeah. So now we think, now that you can see a difference, once you can see a difference, you can begin to tease out to what extent is it the host, which genes in the host, which aspects of the viral genome are actually driving that outcome.
That's really cool.
Yeah. No, we're excited about that.
And there are also viral contributions as well, right?
Exactly.
Presumably. But you're doing more than one genome per cell in these infections, right?
So, yes.
Right.
And so we have no way at this point. Everything is driven by the Poisson distribution.
Yeah.
Until we can light up the genomes in a way that we don't even know exactly what the MOI is for each cell. And to a certain extent, that is going to be to drive certain parameters. But there are only 3 of the phenomenological parameters that we monitor that are driven by viral genetics. We have several that are driven by the host transcriptome proteome.
So in your paper, you show a picture of these microfluidic. They're like smaller than a dime.
Yeah.
They're very small. And you have these specially made for you?
Yeah, so at Penn State, we have a nano fabrication facility. So Microfluidics has been an area of focus in our biomedical engineering and engineering science and mechanics departments for a long, long time. So the overall design of the devices that we use were developed by Steve Quake. And so these are 3-layered devices that you create a mask, and then you use soft lithography techniques to make a mold, and then you pour essentially a polymer into the mold and peel off the device. So we do all of this ourselves.
Mm-hmm.
Yeah. You're welcome.
He's been doing it of late.
Yeah, you do this.
Yeah, and it's a really great experience, though. It's certainly to be able to look at something that's basically invisible and then look at it under a microscope and see all the structure that's there for capturing these single cells. It is a bit like virology. You can't see it with your eyes until you put it under the scope.
Yeah. So you're going to be able to do this at UNC, I presume, right?
Yes. Yeah, so the engineer that we have in the lab right now won't be going with us, but we have a combination of Calvin and Jamie Arnold in our laboratory who have mastered essentially the technology. Another thing that we're doing to try to make this accessible to the community, because this is only as powerful as those who really, adopt this analysis. And so using the devices that we've been using, it's just not very accessible. So we're at our 5th, 6th generation devices now, where they don't require all of these layers. We're able to use a 3D printer to make the molds. And, so that's accessible to anyone. And actually fabricating the devices, this is easy. That's the easy part.
So easy a grad student can do it.
I was just going to ask if you thought that undergrads could do it.
Yeah, absolutely.
And undergrads could do it too.
Okay.
Yeah, so it's the front end of making the actual molds. If you have to use this, dust-free nanofabrication facility, you need specialized training and instrumentation that's just not everywhere. But now that we've been able to transition to the 3D printer, I think this is going to be accessible to everyone.
So in your paper, I love this last 2 sentences. We view the addition of platforms for single-cell analysis of viral infections to be as impactful to virology as single-molecule methods have been to enzymology and single-particle tracking has been to cell biology. The era of single-cell virology is now.
Yes.
Did you write that?
I did.
But it's now if everybody can do it, right?
Yeah.
So
and so that really has been our mission. When we first started this, we didn't know if we were going to see anything, right? And so one of the questions that I always get when I present microfluidics devices is, well, can you sequence the genomes? Can you look at the transcriptome or the proteome? Well, we're in a pneumatically sealed device. No, you can't, right? But if we had spent a lot of time trying to do droplet microfluidics and nothing was there to be seen, then, we would have wasted a lot of time too. So I think that there are advantages to different platforms. And so I think the next generation, which will be really a shift, is to try to adapt droplet microfluidics to this approach as well, so that you can recover the contents of the droplet to see what's happening inside.
Do you think at some point that companies will make these and labs can just buy them?
That's our hope. So once we are confident that this 3D printing and fabrication will work, our goal is to begin to talk to some companies that are creating microscopes. To see whether or not we can create a partnership that will, just allow you to install the chip that you create it's a live cell imaging device. So it's not, most universities have live cell fluorescent imaging microscopes. You would just have to dedicate one that would, take the device. And so it's not that big of a leap.
For me, who's been doing growth curves, single-step growth curves for 35 years, where everything happens at once, it's amazing to see some cells do nothing, some cells in the middle, some cells way out, such heterogeneity.
Yeah.
We shouldn't be surprised, but that will be fun to sort out.
Yeah.
Essentially.
We're very excited.
Different cells, right? Some relevant, like neurons, maybe you could even put in these devices would be fascinating, right?
Yes.
It's cool. And I guess, sounds like you're going to North Carolina.
I'll
right?
be along for the ride as much as he tried to leave me behind.
So you do a podcast called Curiosity.
That's correct, and it's actually on your network. So I'm glad
Yes.
you remembered. I was inspired. I was pretty sick last year, at the end of last year, and I was listening to this other podcast, and they just kept saying this horrifyingly offensive science talk because they didn't know what they were talking about, and it would be really great if we could just, no, let's correct that. Let's make sure that you understand what you're saying. And the thought was, well, if I'm going to be acting in an ambassador position for ASM, as I am right now, a contribution I could do would be to make a science show that's available for everybody. It's totally free, and it doesn't require any specialized background to be able to do. It should be accessible to any taxpayer who funds most of our science, and we are so eternally grateful, and we are so willing to try to share what we've learned if you only take a little bit of time to be able to listen to it. So that was the goal of this podcast, Curiosity. It's spelled just like curiosity, but with an SCI, because it is science related. And you were a guest on it as well. You were on episode 11. So if there was anybody, I know that everybody here is a scientist, but there are things, scientists don't know everything, it turns out. There are topics that you need to review or learn something else about. But if you have a family member that always says, I would love to be able to get into TWIV, but it's just a little out of my reach or something like that, it might be a little bit more in your reach. And you could start with Dr. Raccaniello's episode, too.
What types of topics do you cover?
I love science, so I just really go anywhere. My favorite one to explain, I liked episode 5. It was called How Old Are Ferns? Because I grew up in the Pacific Northwest, just 6 hours north of where we are right now in San Francisco. Ferns look ancient. That's why they were used for Jurassic Park. That's why they were used for Star Wars Episode 6. They bring you to this idea of something old. And it turns out they were developing way before Pangea ever broke apart. And sometimes it's just fun to put that into context with what we can learn. So it's really anything that I want to learn about, but it's also things that I think would engage the public, would make people excited about science, and are diverse enough that it doesn't feel like I'm only talking about polymerases, DNA, or RNA.
Hey, what's wrong with that? So you don't know all these things. You have to learn them.
I need to learn everything. I needed to learn most of what we were talking about today.
Do people write in and correct you?
Absolutely. And also sometimes accuse me of being in the pockets of big insulin, which I am not, but if anybody wants to fund me.
Now, Calvin approached me last year and wanted to put Curiosity. So you're at curiosity.com, right?
Yeah, it's actually ascienceshow.com.
Okay.
A Science Show, because it is a science show.
And he asked me to put it on Microbe TV, which I did, and I was amazed that he was doing one a week. He's a grad student. So I always wanted to ask you, Craig, what do you think about him doing a weekly podcast?
Well, he does that and so many other outreach activities. I mean, I can't complain. I mean, he's great in the laboratory, and I think the contributions
I'm off the hook.
the contributions to outreach are important. I mean, I'm very committed to outreach myself. So, I'm glad that he's doing all that he's doing.
So basically, if he can get the lab work done, it's fine, right?
Yeah, I mean, I don't know where he gets all the time and energy to be as productive as he is in the lab and all of these other things that he does.
He told me in between experiments, he does the podcast.
Yeah, well, I've seen like all the instrumentation, and a lot of the students that he's, interviewed have been in the lab and after the fact, and he's trying to get me to do it. I'm This I think is a foray into the next era.
So Calvin, you're going to go to UNC. You're going to continue to do Curiosity.
That's
right?
correct.
And
yeah.
when you get your PhD, what are you going to do after that?
When I get a PhD, well, let's see. I'll be 75, so I might be thinking about retiring. No, it seems like I really enjoy what I'm doing, and I really enjoy doing bench work. I'd love to go on and do a postdoc and be able to continue that, but also reach out to the public to be able to let them know that science isn't only a mad scientist's work. It is truly a bunch of normal people who don't want to wake up to go to the conference that early in the morning. We're all normal. we do a lot of great work, and you can learn about it pretty simply.
So you'll continue to podcast as long as you can, basically.
I think so, because I'm already trying to do all this learning on the side anyways. What is an atomic garden? I didn't know until I started looking it up, and I thought, that is really cool. I bet somebody would like to hear about that.
What's an atomic garden?
You can expose plants to radiation to see how they mutate. Their genetic elements will mutate, and you can get new funky plants or dead plants.
All right. It sounds like gain of function.
Yeah, right.
I was going to say, it sounds like my gardening skills.
I have a couple last questions for you, Craig. These are fun. What do you think is your most significant contribution to the field of science?
Well, I think there have been two. The first is the recognition that these viral polymerases, ribavirin and other analogs, can be used to target. So you don't have to just, look at chain termination. There are other mechanisms that you can tune fidelity in a way that actually can have an impact for development of vaccines. So I think that those are two very important observations. But then there's one more. I think there's one more. I've been, so bringing Harvoni to market really required the recognition of what the off-target for a lot of these antiviral nucleosides was. And so the discovery that mitochondrial RNA polymerase mitochondrial transcription was the major off-target of most of these drugs actually has led, to all of the companies being able to develop the hepatitis C inhibitors. And so I think that has been the most impactful observation.
Yeah, that's important.
Yeah.
Yeah. So if you hadn't been a scientist, what would you have done with your career?
Well, like I said, I started off thinking I was going to, go to medical school. I can't say I had an idea of, which specialty I wanted to enter. But, I think science was my destiny all along. And so I don't, it would have been medical school otherwise.
Okay, that's good.
So, yeah.
So if you could have dinner with anybody, living or dead, who would it be?
Interesting question.
We just edited out 40 minutes worth of silence for those listening.
Right.
Okay.
You sprung that on me. I don't know.
You can wait till I wrap up, and then before we close.
Okay, why don't you give me a minute to
I have a
think
few
about
minutes
this.
of things to say. Yeah. No problem. I asked the same thing of Brian Cullen a couple weeks ago. He needed a few minutes. But I'll get back to you. All right, so that is TWIV, episode 555. Most of you probably listen on your phone or your tablet using a podcast player. We'd like you to subscribe. That way you get every episode automatically, and we know how many people are listening. It's really important to us to be able to raise money. So please subscribe. It's free. And if you really like what we do, consider contributing. You can go to microbe.tv/contribute, and PayPal or Patreon, you can give $1 a month. And I always like to say, what are you going to do with $1 a month? You can't do anything with it by itself. No coffee, you can't buy anything. Give it to us, and in return, you will get not only TWIV, but 4 other science podcasts, TWIP, TWIM, TWIVO, and Immune. All factual science truth, as far as we know. Science podcasting for $1 a month. So we'd really appreciate that. And as always, questions and comments, twiv@microbe.tv. My guest today from Penn State University, Craig Cameron. Thank you so much for joining us today. And my guest co-host, also from Penn State, Calvin Yeager. Thank you
so much for having me.
Pleasure. Brianne Barker is at Drew University on Twitter. She's BioProf Barker. Thank you, Brianne.
Thank you. It was great to do my first in vivo TWIV.
And Calvin, you're on Twitter as well, right?
Yeah, I'm on Twitter at Cal Yeager. I don't usually get called Cal, but now I have been through all this. So go ahead. We're friends.
Cal Yeager. You're not on Twitter, right?
I am, yeah.
What are you on Twitter?
Cameron Lab PSU.
Cameron Lab PSU.
Yeah.
Oh, you're going to have to change
We're going to have
it.
to change that. They tell me it's possible.
Yeah? Okay, good. I'm Vincent Racaniello. You can find me at virology.ws and Prof VRR on Twitter. I'm also on Instagram, YouTube, all the same, Prof VRR. I want to thank ASM Microbe for allowing us to do this again this year. And I also want to thank ASM and ASV for their support and the introductory music by Ronald Jenkins, ronaldjenkins.com. Did you come up with
Yes, I
some?
did. Gertrude Elian.
Okay.
She was a Nobel laureate, of course. She's credited with, the development of the first antiviral nucleosides, the herpes drugs.
Cool.
I think it would be cool to kind of see what was in her head and what she thinks about what we're doing now. And
Yeah, it's always cool to get people who were around a while ago and say, hey, here's what we're doing now,
Yeah,
right? That would be fascinating. All right, let me give away some T-shirts here. Do we have any TWIV fans? We got one here.
everyone.
Any others? You're a TWIV
Oh my
fan?
God, he's throwing it across the entire room. His arm is so strong.
We'll go back row. Nobody back there is a TWIV fan?
Oh, now
Let's see if I can reach. There you go. Oh, one more. Look at that. All right. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWIV is viral. Thank you.